Alright, okay, so hello dears. Ayan, so welcome now again to the part 2 on our lecture on, again, complement fixation or on the complement system. Alright, and for this part, we're now going to focus our attention on the activation and the work and how the classical pathway works. So classical pathway lang ha. Alright, the rest of the pathways si Ma'am Teddy na. Okay, sa lecture na. Alright, ayan, so when you say complement fixation guys, basin mo ka-ask mo anong fixation. Complement fixation, when you say fixation, complement is being used up, all right? Or the complement is being used for, or the pathway is being activated. Muna siya points of fixation. So, na-fix siya ba? Meaning, na-fix siya sa antibodies ba? Or dito sa bacterial membrane? Ganun. So, muna yung point sa complement fixation. All right, okay. Now, for classical pathway, again, we'll start, of course, with your recognition unit, which is the C1QRS. All right, so ang C1Q, good guys, ang mubind sa imuhang antibody, di ba, sa FC portion. Now, once the C1Q is mubind na siya, the next protein, C1R, C1S, ma-activate po sila. And then they form now katong a strong, parang complex in a way, which is known as your C1QRS. Okay, C1QRS. All right. Now, this recognition unit, it then activates, next is the C4 protein. All right. And what it does is, it cleaves C4, alright, so C1QRS, okay, iyahang i-cleave ang C4 into C4a and C4b, okay? Alright, then recall, di ba, unsa man to na letter lang ang mabilin, kaya siya ang heavier, siya po na enzymatic activity, so C4b. So, kanira ang C4b. Now, the C4a, it goes out of your serum or goes out into the environment. Alright, now C4b, it then activates your C2 into C2a and C2b. Diba? Alright? And again, it is only in this category, diba, na C2, ang C2a ang magpabilin. Alright, siya ang mustay. Char. And ang C2b ang mawala. Alright, so siya ang mustay, C2a. Now, C4b and C2a, they now join, saan ako mabutang, diri na lang. They now join to form a complex known as C4b, C2a. Alright? Okay, and this is now what we call your C3 convertase. Okay, so sa may point sa C3 convertase. By the name itself, C3 convertase, so this complex now activates, di ba, activation unit, your C3. By the name itself, kaya siya man ang C3 convertase. So iyahang bungkagon ang C3 into, still the same, C3a and C3b. Alright? Okay. Ayan. C3a, C3b. Now, again, as mentioned, kay since C3 man siya, unsa may mawala? Ang C3a. Magpabilin ang C3b. So, mawala ni siya, pabilin ni siya. Now, this C3b, it now combines with your C3 convertase to create your C4b, 2a, 3b, okay? Which is now known as your C5 convertase. Okay, C5 convertase. And by the name itself, basta convertase gani, meaning iyang i-activate ang C5. So your C4b, 2a, 3b, C5 convertase activates C5. Alright? Bungkagon niya ang C5 to C5a and C5b. Gets? So again, still the same. Since C5 naman siya, dili man siya C2, kung sa may magpabilin, ang A or B? Ang B. So, ang A, mawala na pod. Alright? And C5b, alright? The C5b then will now start the formation of the membrane attack complex. Dito siya mag-start. So, C5b then, C5b, yung i-activate ang C6. And then, the C6 activates C7. Then, still the same, padayon na C7 to C8. And then, finally, C8 to C9. Ayan. So, that's it. Diba? Okay, C7, C8, and C9. Finally, this C5b to 9, okay, this is now known as your MAC, or your membrane attack complex. Okay? Alright, and this membrane attack complex then goes, or then serves as your, or then leads the eye to the cell lysis. Okay? So, in a nutshell, muna siya ang function or how the classical pathway works sa complements. Okay? Alright. Now, please take note lang, di ba? Very confusing siguro here, no? Akong gibutangan og line here, guys. sa taas sa C4b, 2a, 3b, and actually put diri sa C4b, 2a, it means that it is the active part na. Siya ang active enzyme. Okay? Or siya na ang convertase. Okay? Alright, so na-align sa taas. So in a nutshell, wow, Mart, I'm impressed with myself, no, in a way. Alright, so C1 dyan siya mag-start. Okay? So in a nutshell mo ni siya, and mupod ni siya. So, I don't know kung asa. Try your best daw if to write by yourself kung sa functions, ay like sa stages, okay, sa different proteins. Try to, now without looking, after na ninyo watch, ani, alright? To just test yourself if nakasabot ba jud or ano. But I will understand, I would understand if at first pag yun medyo confusing, because sa ako pong when I was a student po, medyo confusing po siya. Alright, okay. But kani siya guys ha, dili squares ang hitabo inside your body. There's a picture in your Stevens, wala na ako nabutang here, na giun sa siya pag-create ang membrane attack complex. So this happens on the surface of your bacterial membrane or on the pathogen. Okay? So there are different proteins, it pass, like example, kaning C4a, it's a protein na mupas sa C1QRS. Pagpas niya sa C1QRS, mabulag siya, and then mutapot tong C4b, sorry. The C4b mutapot siya sa surface sa membrane, parang inana. Alright, na picture sa Stevens inyo na book. Okay? Nalimot ko butang here. Sorry. Alright, okay. But again, this is in a nutshell, just to draw lang, okay, how the pathway works. Okay? Alright, ayan. So erase. Erase. Alright. Okay. Ayan. Okay. Now, I have in a way, mnemonic siguro that can help you para lang sa number. So, we'll start first with, again, 1, 4, 2, 3. Ayan, 1423. Okay? That's how I remember it. 1423. So, dira ka mo start sa C1. Then, of course, you activate ang C4. And then, the C4 activates C2. And then, finally, the C3. Now, for the convertase, for C3 convertase, always remember C4B, 2A. So, mag-alternate ang B og A. Okay? And for C5 convertase, you have C4B, kapira, 2A, and 3, alternate Japon, B. C4B, 2A, 3B. Now, take note, this is the convertase, C3 and C5 convertase of the classical pathway. Because in your alternative pathway, na siya'y lain. Lahi iyang C3 convertase o C5 convertase. Alright? You'll have that in your lecture pa rin. Okay? But for classical pathway mo ni siya, C3 convertase and C5 convertase. Okay. Now, just some information, which again, i-discuss mo sa lecture, but ako na lang introduce here, yung mga important stuff. C3, okay, the protein C3, guys, is the most abundant, okay? The pinakadaghan na complement protein in your body, okay? Please take note, C3. Now, C3 also is considered to be the pivotal point, okay? Pivotal point or the point kung asa mag-merge ang tulo ka pathways, alright? Because you'll know in your lecture na once ang the classical, the alternative, and the lectin, asa sila mag-abot na protein? Sa C3, okay? So, ang tulo ka pathways dito mag-abot sa C3. So, all of the pathways, they all have C3, or they use up C3 in their activation, okay? So, again, ang question is, asa na point, or what protein is the pivotal point, or what complement protein is the pivotal point, or the point of merging, siguro, limot, sayo pa akong term, but basta dira ang point na mag-meet silang tanan, ang tulo ka pathways, kung sa ito na complement protein, C3. Alright, that is why, since ang tulungan ka pathways mag-abot sa C3, deficiencies in the C3, if na kay C3 deficiency, alright, C3 deficiency, meaning wala kay C3, that's why it results to severe infections, okay? So, siya ang pinaka-severe and the most serious, okay? If na ay deficiency. Because again, of the reason that C3, siya ang merging point, okay? Sa imuhang tulo ka pathway. So, if gamay mo C3, could be na ang classical pathway dili magpadayon, ang alternative pathway di po magpadayon, and then your lectin pathway also will not push through. Therefore, walay may tabong sa lysis. So, magpadayon na imuhang antigen. Okay? That is why C3 deficiencies, the most serious and results to severe infections yun. Okay? Recurrent infections, meaning balik-balik. Alright? Okay, so again, C3, the most abundant complement protein. in the blood or in the serum. And again, the pivotal point of all the three pathways, or this is the point where the three pathways meet, okay, mag-converge sila, all right? And again, the most serious when it comes to deficiencies, all right? And C2 naman is the most common deficiency, okay? The most common deficiency, ang C2 na protein. And C9 is the deficiency without any disease. Okay? Alright. So meaning, ang C9, if mamubo siya or mag-deficiency ka sa C9, wala rin disease na may tabo. Because again, your C9, multiple copies man to siya, di ba? So, ang gingon dito na C9 accelerates the lysis of your cell, but if kulang siya, magpa-dinner gyapon og lysis. Okay? So, munang dula kayo disease or walay disease known if na ay deficiency with C9. Okay? Alright. So, please take note guys ha, lumalabas sa boards. Very important din na mga information. Okay? Alright. Ayan. Okay, I hope na gets lang. But sige lang, try your best. Slowly. Okay? Alright. Ayan. Now, we go now to the test. So, like, why are we talking about complement system? Because again, there's a test for that using complements. And because again, complement fixation occurs after antigen and antibody combination, then we use this test or we exploit this principle to determine either an antigen or antibody in our body, okay, depending on the disease, okay, depending on the antigen that we want to determine. And true enough, this is used before in testing for syphilis, okay? The Washerman Test. The Washerman Test is considered to be the first serological test for syphilis. Please take note, guys, lumalabas sa boards. Washerman, the first serological test, if I'm not mistaken, used to detect syphilis, okay? All right, we'll have that in our syphilis serology, okay, later, okay, in the semester. All right, and this technique also has been used to determine or detection of viral, fungal, rickettsial, and protozoan diseases. So it has a lot of uses, all right, complement fixation. All right, now for the test proper, it has a two-stage process. Number one is the test system, which consists of the antigen and antibody, all right? One is unknown, and for our example, the unknown is the antibody, all right? Okay, so that's the test system. And we also have the indicator system. When you say indicator, siya ang system na mo indicate. Wow, Mark, explain pa ka. It's a system that lets you know the ayan, indicates if naabadyoy ang antigen or naabadyoy ang antibody or antigen or wala. Okay, and for our indicator system, it uses sheep RBCs with hemolysin. When you say hemolysin, guys, it's an antibody, all right, that mediates or promotes hemolysis. Okay? So, ang sheep RBCs nimo, coated na siya daan with hemolysins or antibodies that promote hemolysis. Okay? So, kani siya, manufactured ni siya. All right? Okay. And of course, the sheep RBCs will lyse in the presence of complement. Diba? Because again, the cell itself, your sheep RBCs, na siya antibodies na nakatapot. Alright? And diba, your complement will attach to the FC portion. Okay? Alright, ayan. Alright, now for the procedure, first is of course, the patient's serum is heated to 56 degrees Celsius for 30 minutes. Ayan guys, so this, please take note of this ha. This is the part where, joke lang, this is the specifications for patient inactivation or heat inactivation, okay? Because again, your serum, diba, as mentioned, complements exist there, normally, diba, in our body, with or without infection, okay? Now, because we don't want that to interfere with our test, of course, ato siyang i-heat inactivate. Because again, your complement proteins are heat labile, alright? So ato siyang walaon, alright? Ang complement proteins daan na naa sa imong patient serum because we don't want any interference from them. Okay? Alright. Because apil sa atong reagents ang complement. Okay? From another source. Alright? And we don't want, again, the complements that are normally found in the patient's serum to interfere with our test. Okay? Alright. Ayan. Now, we make dilutions. Ayan. So, mag-dilute na punta. No? Not necessarily serial dilutions, but you can perform later, di ba? Ako i-explain po. You make dilutions of the patient serum, and again, para, di ba? Purpose of dilutions para again equal, no? Para equal ang antigen antibody na way. Okay, para optimal ang pag-react. Okay, all right. Ayan. And again, you add a measured amount of guinea pig complement. Ayan, so the complement mismo is a reagent. Okay, so hence the reason why we inactivate our specimen. All right? Okay, because we don't want, again, any interference from the complements found in your serum na. All right? Okay. And we add your indicator system, the sheep RBCs coated with hemolysins. We incubate, centrifuge, and then read for hemolysis. Diba? In your blood bank, you have already been taught, siguro, or i-teach pa, on how to check for hemolysis. No? Ayan. And positive siya for the antigen or antibody unknown na gipangita if there is no hemolysis, and negative if na ay hemolysis. All right, so and we also use titer if you want to determine the titer. It's the highest dilution of the patient's serum without hemolysis. Okay, so you'll be wondering, sir, anong without hemolysis ang positive or negative ang with hemolysis? So don't worry, yan ang next slide. So we'll discuss now the test system. Okay. So we have the reagent antigen. Okay, as mentioned, it's found in the reagent. So it's manufactured, no, gikan siya og lab or whatever. Ayan. So it's part of the reagent. Plus your patient sample, so ito siyang i-combine. So this time, since manufactured man ang antigen, so therefore ang atong unknown is antibody. So we're looking for antibody. If the patient has this antibody ba to this particular disease. Okay, example, ang reagent antigen here is syphilis na antigen. Okay, so we're looking for if ang serum ba sa patient has antibodies to syphilis. Okay, all right. Ayan, just an example lang. So we'll start first with the serum with antibody, so meaning the patient has the antibody. So of course, since the patient has the antibody to this specific antigen, katong para sa syphilis, mo-combine sila. So they have already a complex form. All right? Okay. If without the antibody, of course, so the reagent antigen is freely floating. Walay nahitabo. Kaya wala may antibody na ni-bind niya. Okay? All right. Now, we add the complement. Okay, so what do you think will happen? All right. So since your antigen-antibody combined already, there is a complex already na form. The complement, di ba, guinea pig, which is still another reagent. So since na may antigen-antibody complex, di ba, recall, di ba, ma-trigger siya of the AG antibody complex, classical pathway. So they now combine. So this is the point of complement fixation. When we say fixation, the complement has been used up already, okay, by the antigen-antibody complex. All right? Whereas if walay antibody here, di ba? So walay mahitabo. The complement is free, wala siya na used up, so meaning it's not fixed. All right? So walay complement fixation na hitabo. All right? Gets. Now after this, di ba, take note of this outcome. We then add your RBCs, indicator system. Now, di ba, recall that your RBCs are coated na with antibodies. All right. Now, serum with antibody, let's say, adan mo siya sheep RBCs with hemolysin, so what do you think will happen? Of course, the complements has already been binded or have already been binded to the antigen-antibody complex. Therefore, wala na complements na mutapot sa imuhang sheep RBCs with hemolysin. Therefore, all right, di ba? So, wala may complements na nitapot diri. Alright? So, please take note of that. And then, ato siya i-compare with the second one. So, free naman ang complement, di ba? So, pwede pa siyang mutapot sa sheep RBCs with hemolysin. Right? So, therefore, the outcome is, since, again, first one, ayan, wala na may complement na pwedeng mo attach diri. So, dili matrigger ang hemolysis. Therefore, no hemolysis occurs. Okay? And the second one is, of course, no hemolysis, which indicates positive test, meaning the patient has the antibody, okay, to the reagent antigen, so meaning na siya antibody example to syphilis, all right? And lastly, of course, since free raman ang complement na mo-bind sa imuhang sheep RBCs with hemolysin, so therefore, iyahang i-bind tong antibodies na nakatapot sa sheep RBCs, which then promotes lysis. So therefore, that's the negative test, hemolysis. Okay? Nagets lang? Alright. It means that your patient doesn't have the antibody, okay, to this antigen. Kaya wala man sila nagbindaan, di ba? Okay. So meron siya agglutination inhibition in a way na principle. Alright? But this time, hemolysis ato ang ginadetect. Alright? Okay. And so I hope nagets lang. Okay. Alright. And for, yes, just some precautions. Number one is, again, it's very important to heat inactivate your serum because, again, of the reason that your serum, with or without infection, contains complement proteins. And we don't want any interference from them. Okay? Because again, imagine, no? Your serum has already complements. Then pag-add nimo sa imuhang reagent antigen, sa test system palang daan, wala pa nimo gibutak ang sheep RBCs, pwede nang mag-lyse, no? Or may-use up na tong antigen or antibody. So wala na yung mahitabo sa indicator system. It could lead to false negative, false results na lang. Okay, wait, wala ko think about that. But that's the point, okay? We don't want any interference from the serum mismo, okay? So dapat important yun ang heat inactivation. Again, how? 56 degrees Celsius for 30 minutes. Please take note, ayan. Stored serum becomes anti-complementary. So if you store serum, pwede siyang anti-complementary. So what it does is pwede niyang i-attack tong complement na reagent. So therefore, leads to erroneous results pa rin. Alright? Okay. Elaborate QC standardization required, of course, because we want that our results are validated, no, they're accurate. So we need to perform rigorous QC. Alright? Especially that we use mga sheep RBCs, no, guinea pig complement, which are prone to mga deterioration and if improper ang storage and handling and all that. Alright. Okay, more useful for IgM antibodies, again, as mentioned, because, again, IgM is the largest, it's big, so it's really best for complement fixation. So, mas okay yun siya gamitan for IgM. Can do serial dilution for semi-quantitation. Yes, so, example is a test known as your CH50. So, stands for complement, and H means, this is actually hemolytic titration. So, what it does is you detect the 50%, no? Yes. Okay, you detect the amount of serum required, serum of the patient, no, required to lyse 50% of the concentration, all right, of your solution. Okay, all right. So 50% again, you detect the amount of serum from the patient, all right, required to lyse 50% of your sheep RBCs na given, all right, katong naay hemolysin, all right, because if naay deficiencies in the complement proteins sa imuhang patient, ang imuhang 50% na dilution or na titer will go down or will become 0 because dili siya makalyse dayon by 50%. Okay? Alright, so by 50%, so imo na siyang i-plot. So you have mga different dilutions here, like 1:20, 1:40, 1:80, ganun. And then you have different tubes, no, of different dilution. And then you determine the 50%. So let's say ang 50% kay 50% of the hemolysis, meaning 50% of the concentration of the sheep RBCs that were there were already lysed by your patient serum. So let's say sa 1:80 dilution, no? So ayan. So your titer therefore is 80 IU per ml, example. Ang 50% na nahitabo ang lysis. Because again, if there are deficiencies in complement proteins, mugamay siya. All right? So makita nimo. All right. Okay. And then next is, of course, PEG enhances rate of antibody uptake. So, katong mga ato ang enhancers. Polybrine also brings cells closer to for lattice formation, which again promotes the antigen-antibody complex formation. And AHG can be used also to detect sensitized RBCs. So, if you want to detect if the sheep RBCs still contain antibodies or hemolysins, you can add AHG and then look for agglutination, which means that nadya po yung antibodies na naka-attach sa sheep RBCs kaya na-bridge man siya by the AHG, alright, which then forms the agglutination. Alright? Okay, ayan. And that's the end for our complement fixation na test. So it's quite short lang din naman, pero medyo maraming basin TMI. Alright, so try your best to do it slowly lang, guys. Yes, slowly lang good for the first time. Huh? Alright, okay. But yeah, so that's all. I hope na gets lang. Medyo ano siya, no? Again, you'll be introduced to the different pathways pa and their complement regulations, the proteins that regulate complement system in your lecture. Alright, so ipapaubaya. Yes, ko na kay Ma'am Teddy yan. Alright, so again, mura to sa tanan for this lecture, guys. No? So I hope you appreciate it and you learned something today. Alright, and if you have any questions, feel free to chat me or chat sa atong group chat, no? And I'll see you in our next pre-recorded lecture. God bless, guys, and keep safe.